Thank you for joining us today. We're so happy you could join our webinar. My name is Nicole Navratil. I'm the head of marketing here at BASI Research Products, and I have the great pleasure of introducing our speakers today. This is the second webinar in a series of electrochemistry webinars that we are hosting. You can learn more about our future webinars on our website at basinc.com/upcoming webinars. And that also includes links to our past webinar recordings. Today, we're going to learn about how spectroelectrochemistry methods can help provide deeper insights into many research questions from bioelectrochemistry, energy storage, to electrocatalysis, environmental analysis, and more. And our speakers aim to illuminate both the fundamentals and the implementation of spectroelectrochemistry techniques in key application areas. And I want to just go over a few things before we dive into the fun stuff. Please note this webinar is being recorded and a link will be provided to everyone who's registered. It'll also be available on our website. We do ask that you hold questions until the end, so we will have a Q&A session at the end, but there's a Q&A button here. Should be at the top of your screen. Feel free to add your questions whenever they come to mind. That way they're there, and then we'll go over them during the Q&A session. Please use the chat function. I'll be monitoring that in the background. So if you have any technical difficulties, issues like that, please feel free to use the chat function to communicate with me. And once we join our interactive Q&A, you can keep adding questions to the Q&A and we'll keep activating them and asking them as we go. There's also the option to raise your hand if you prefer to speak directly. And there's also controlled captioning if you want to use that as well at the top in your menu bar. So let's get to the good stuff. I'm very excited to introduce our invited speaker, Dr. Megan Lazorski from the Metropolitan State University of Denver. She also holds a joint appointment with the former National Renewable Energy Laboratory and recently renamed the National Laboratory of the Rockies. Her research focuses on photophysical investigation of structure-function relationships with specific experience in developing novel inorganic photosynthesizers and 3D spectroelectrochemical instrumentation. She currently serves as the PI on a $2.25 million DOE collaboration, as well as a co-PI on the NSF-funded Center for Sustainable Photoredox Catalysis. Beyond her scientific contributions, she is dedicated to mentoring and community building, and she founded the MSU Denver Post-Baccalaureate Bridge Program, and we're very excited to have her here with us today to share her experience and provide some foundation and the fundamentals of spectroelectrochemistry techniques. So Dr. Lazorski, take it away.
Right. Well, thank you so much, Nicole. I'm delighted to be here. I feel like this is such an honor and what an excellent opportunity to talk with you all today about a topic that I find fascinating and exciting. That is spectroelectrochemistry. So we'll just get right to it. Today, I outlined this talk in the form of learning objectives because I am a professor and that's how I think anymore. So I'm going to kind of outline for you some of the brief history of spectroelectrochemistry and kind of discuss why we are interested in increasing awareness about this awesome technique. I'm going to describe some of the basic principles and uses of spectroelectrochemistry, as well as demonstrate the versatility of this technique. I will relate.
I'm sorry to interrupt. I don't think your slides aren't showing yet.
Oh, they're not? Oh, gosh. Okay.
Sorry about that.
No, it's okay. I'll go back to the beginning. I thought, all right, cool.
Perfect. No, they are.
Can you see them now? Okay, perfect. Yeah, okay. Well, that's my title slide. Not super fun, but I like my second slide because it's got, dark side of the moon. So I was just going through the first 3 bullet points here. And after I talk about the versatility of the technique, we're going to kind of relate instrumental design of at least transmission-based spectroelectrochemistry setups to their function, and discuss recent advances in SEC applications, as well as some of the barriers that exist and some of the solutions to those barriers. All right, so kind of a really brief history of spectroelectrochemistry. It was developed in Kansas, at University of Kansas, by Dr. Theodore Kawana in the 1960s. I think his first publication was in 1964. And since then, there was a really steady growth in the interest and publication about this topic until about the 2010s. And then we've got a kind of sharp decline recently, not super sure why. So it seemed like a good time to reintroduce this versatile technique to the public, and just demonstrate how this method is used to understand changes in molecular, thermodynamic, and kinetic properties of electrochemical processes using spectroscopy. Two of my favorite areas of inquiry are spectroscopy and electrochemistry. So it's a excellent place for me. So what is spectroelectrochemistry? If you've never heard of this technique, basically it is a technique like that. It doesn't have a fancy name that's meant to confuse. It's very straightforward. It uses a spectroscopic method to study a redox process using electrochemistry in situ. So if we are generating redox species in situ with a working electrode, we're able to use a spectroscopic technique to see the changes in the molecular, thermodynamic, and kinetics of either a reaction process, an electron transfer that's happening due to light, or many other applications. So basically, we use the spectroscopy to observe the oxidized and or reduced forms of the analyte. And then the spectroscopic signal provides information about the energetics of the redox species, redox-induced structural changes, rates of redox reactions, and much more. So as I said, this is not a fancy name. It's pretty straightforward, spectroelectrochemistry. And we shorten that to SEC because it's less of a mouthful, and it takes up less of the page. So spectroelectrochemistry is especially useful for species that are hard to synthesize or isolate in solution because they're transient in their nature. So something that requires an in situ generation process through an electrochemical technique. Also, if something is hard to isolate or it's unstable for these same reasons, but can be generated electrochemically, this is another way to study the properties of those kinds of materials without having to use chemical reactants that can have unnecessary or unwanted side products and things like that. One of the criteria, though, for these complexes is that they need to be able to be produced easily in solution via electrochemistry, or it doesn't have to actually be in solution anymore. You can also do solid state reactions, but you have to be able to generate the redox species easily with an electrochemical setup in situ. So some of the use, there are actually many uses of spectroelectrochemistry. Basically, if you can imagine a spectroscopic technique that could be coupled to an electrochemical process, then you can make a spectroelectrochemistry setup that could measure that as long as you get all of your parameters correct. So it's very versatile. It's used to probe a ton of different chemical and physical phenomena, and can be used for a variety of different studies that include any type of redox active species. And as I stated before, it's most often used to observe species that are not observable by other means under normal conditions, things that are hard to synthesize, isolate, or are unstable. So kind of the first high-level example here is a really interesting one to me and my research. because it involves a non-heme first row transition metal oxo complex. And these complexes, particularly in the +4 oxidation state, are pretty unstable, and they are hard to generate in situ, or they're hard to generate with chemical means, and then also be able to actually characterize. So in this paper, someone was able to use a high-speed Raman CV technique with an electrochemical pulse. And so they were able to actually obtain the redox potential of the iron 3, iron 4 redox couple, which is notoriously difficult to obtain with these complexes due to this word is always hard for me to pronounce, comproportionation and surface adsorption kind of competitive kinetics. So this high-speed technique enabled the electron transfer for the 3-4 couple to be faster than the adsorption and comproportionation processes, and enabled them to not only determine that E1/2 of the 3-4 couple, but it also enabled them to determine that the redox or the oxo complex could be generated in situ without the use of a chemical oxidant at all, which is a kind of source of frustration for these types of complexes. So this is one demonstration of how SEC can be used to investigate transient states that are difficult to characterize by other means. Another interesting kind of older paper, but I love this one because I teach inorganic chemistry, the perfect place for spectroelectrochemistry to come together because you use structural, you're looking at electron transfer, and you're looking at spectral techniques, and often electrochemistry, everything comes together in inorganic for me because, you know, I'm obviously not biased at all. But this is a little bit of an older paper, but I thought it was really interesting because using spectroelectrochemistry, and in this case, they used a combination, I believe, of IR and UV vis spec-EC. They were able to see a transition from an eta-2 to an eta-1 coordination mode to this tungsten center. And they were able to watch this isomerization happen through the spectroelectrochemical technique, demonstrating that you can use SEC to probe redox-induced structural changes, as well as just transient states that are maybe not as stable. So these are, this is a transition between two stable species, but the isomerization was difficult to capture by other means. Another really interesting application that I've seen over time is that people have used spectroelectrochemistry with confocal microscopy, where they're able to visualize single redox processes happening. And we all know that almost all chemical techniques study the properties of bulk, large numbers of analyte molecules simultaneously, so we can only describe bulk behavior. But in this instance, they were able to use this confocal microscope spectroelectrochemistry setup to allow the detection of single fluorescent molecules. So the differences between single molecule behavior and bulk behavior could be differentiated. And there are many important issues or instances where the issues of mass transport are concerned that could impact these differences between the bulk and the single molecule behavior. So this was a really interesting application of spectroelectrochemistry using confocal microscopy for single molecule visualization. Another interesting recent example is using spectroelectrochemistry with FTIR to elucidate the reaction mechanism where this ruthenium nitro complex is able to is useful for delivering radical nitro group or nitro radicals. Sorry, my brain was glitching there, for therapeutic applications. And in therapeutic application, this requires a really controlled delivery and a controlled concentration of the nitro radical. To do that, the mechanism of nitro radical delivery has to be better understood. So in this study, they started with this pyridine appended ruthenium complex that is trans to our nitro group that is going to essentially lyse upon excitation. And that is the delivery of the nitro radical mechanism. And so what they found out is that this mechanism really relies on the cis-trans isomerization, and the trans effect can actually be used in this case to deliver the nitro radical in a more controlled manner, as they started to understand that when these isomers are generated, they're generated via this outer sphere electron transfer reaction that then triggers this chain electron transfer reaction that then releases the nitro radical into solution. And so this is just another example of how FTIR SEC can be used to elucidate reaction mechanism. And this is only a small example of the many, many different ways that SEC can be used to understand different systems. And so I just gave you an example of confocal microscopy, SEC, UV-Vis, SEC, FTIR, SEC, surface enhanced Raman, SEC, and just regular IR, SEC. It turns out that there are many, many other ways. Basically, like I was saying before, almost any spectroscopic method that you can think of, you can pair with an electrochemical technique, as long as you can get the instruments to talk to one another well, and the experimental parameters to be set up in a manner that will allow you to get both the electrochemical and the spectral data. But this is kind of an example of some of the most common ways that SEC can be used. They can be used in transmission cells, and this is common for UV-Vis and IR applications, as well as reflectant cells. Some other absorption spectroscopies that are often used, or are maybe not often, but are definitely used, and there is literature precedence for the use of SEC, are SEC with EPR, NMR, X-ray absorption spectroscopy, and many other techniques. And so these all kind of rely on this setup where we have an electrode and we have light interacting with our analyte in some way, whether it be a transmission mode where the light passes through the analyte medium, or a reflectance mode where we have a reflection off of a back contact, which is our electrode. And apologies, my transparent electrode is supposed to be at the front of this little internal reflectance cell, but it got shifted there. You can also use SEC with scattering and luminescence techniques, which we did see in the uses slide. We saw that we could use surface enhanced Raman for an SEC application. You can also use fluorescence and other emissive types of techniques. And really the big deal here is the way that you set up the cell, and the way the cell or the way the spectral data and the electrochemical data and the instrumentation talks to one another. Those are the things that are kind of the biggest things to consider when you're trying to set up a spectroelectrochemical experiment. And so I'm going to use the UV-Vis spectroelectrochemical setup as kind of a standard, because it is kind of the first application of spectroelectrochemistry, and it's kind of the best, it's one of the best ways to start your path in understanding this technique. So we're going to start here and kind of demonstrate some of the critical components of UV-Vis SEC so that you can see some of how these, the design of these cells really impacts their function. So on the left-hand side of the screen here, I have just a little figure of some of the components of a typical UV vis cell that we're going to set up for a spectral electrochemical application. And on the right-hand side, I have kind of a side and a front view of what the cell looks like when it's all assembled. And we're going to walk through this here. All right, so of course we need to let the light through. So we need a optically transparent cuvette, which we've got here, and that enables our light beam to pass through our analyte, just like any regular spectral technique. We also need a reference electrode, of course. Right here I'm showing a reference electrode that's a hollow body, and then you can fill it with a non-aqueous electrochemical solution. Typically, we use silver nitrate in acetonitrile. That's a very common one, but any type of reference electrode could work as long as you can get it to as long as you can get the setup to work with the rest of the components that are necessary for the whole cell to function. All right, on slides, there we go. All right, the working electrode is a very important part of the cell setup, particularly for UV vis spectral electrochemistry, which is a transmission mode of spectroelectrochemistry. So you would imagine that because we are doing electrochemistry and we are doing a spectral technique where light has to pass through a cell and be detected by a detector, we have to have a very special type of electrode that enables light to pass through, right? Where we have two different examples here. The wire mini grid is a very common, it's a wire mesh. It's a very common type of electrode setup. Another common electrode setup that has become more common in the last maybe 10 years is a screen printed electrode, which features a honeycomb kind of architecture here where the light can pass through the white circles, and the screen printing deposits a gold or platinum metal onto a ceramic body that then enables the electrochemistry to happen around these the outer parts of each of these holes. All right, and then we, of course, need our counter electrode to complete our electrochemical setup. And all of these are integrated into our cell, and you can use just like a normal platinum or gold wire, or on the screen printed electrodes, it's common for the counter electrode to be integrated as a planar electrode onto the screen printed setup. So when we set this up, our working electrode goes down into the path of the light beam, and our analyte mixture is, I should say, the working electrode is submersed in the analyte mixture in a small path length cell where the light beam passes through, is probably a better way to describe it. We've got our reference electrode that also has to be, submerged into the analyte mixture so that we can reference our voltage. And then we have our counter electrode to collect our current that is also submerged in the mixture. The kind of image here shows a lot more solution than you typically need, but as long as the some portion of the electrode is submerged in the solution, you can get your electrochemistry to happen. And as long as your solution is above the path of the beam by a decent amount, and both of those things are satisfied, then you can get your spectroelectrochemistry to occur in this type of architecture. All right, and like I was saying before, we can set up a spectroelectrochemistry device with a variety of different spectral applications. You can also do different types of electrochemical experiments in these different modes. But since you are using different spectroscopy techniques, that means that the energy of the light is different of the excitation that's occurring in each one of these spectroscopies. And if the excitation energy is different, you're probing a different either structural change, a different degree of freedom, and you're also working on a different time scale for a process to occur. And all of these things are critical to understand to set up a functional SEC cell. All right, and so this necessitates different instrumental designs and different instrumental response functions. And so there has to be a precise synchronization between the spectral instrumentation and the electrochemical instrumentation, and the time scales have to fit for the process that you are trying to observe. All right, so here is just an example of 3 different types of cells, and you can see the UV vis cell that I just showed you, and how that is being kind of extrapolated in different architectures and different designs here to modify the spectro-electrochemical technique to do a different type of spectroscopy. And kind of the take-home message of this slide is that because we are using this combined technique, the nature of both the spectroscopy and the electrochemistry influence the outcome, and that makes the cell design extremely important. So here you can see, I think this luminescent cell is a good way to visualize the difference in cell design, as is the near IR cell, but I think this one is very important because as anyone who does any type of luminescence or emission spectroscopy here knows that your excitation light goes in and through the sample, but you don't detect the light that is transmitted through the sample, you detect the light that's emitted, so you have to detect at a 90-degree angle. If you're detecting at a 90-degree angle, that is going to change the way that you set up your equipment, and that's going to be much different than the way that you're going to detect in a UV vis setup. All right, so some of the requirements, general requirements that you have to think about when you're setting up some kind of spectroelectrochemistry experiment is that you have 2 different techniques here, spectral and electrochemical. Whenever you're doing electrochemical technique, you obviously need to have a electrolyte. So that is going to influence what you can do. And different types of spectral techniques require different types of solvent systems. For example, if you're using a microwave technique, then you're not able to use very high polar solvents because of the amount of heat that's generated through the microwave excitation. So you have to think about how the electrochemical requirements and the spectroscopy requirements are going to work together to facilitate the process that you're trying to see, right? So you've got to have also a redox species that's easily formed through an electrochemical process. And you want to also have electrochemical processes in your analyte that are well separated enough so that you can probe one at a time, or at least be able to deconvolute them from one another. And typically this means that you want individual redox couples to be approximately greater than or equal to approximately 100 millivolts. You can push that limit, but you're trying to have, enough separation between the redox couples in the particular analyte so that you don't have too much overlap and have to do a lot of analysis to deconvolute the signals. And you also need to make sure that, of course, that the redox or oxidized species that you're trying to detect has an actual signal in using the spectroscopic method that you are trying to use. So all of this together, then you now have to think about how well am I able to synchronize the equipment for the spectroscopy and the electrochemistry, and are they able to talk fast enough that we're able to see these processes happen in real time? So are we able to detect the kinetics of the or are we able to detect what's happening fast enough to actually capture the kinetics of a particular reaction or a particular transformation with the setup that we have? All right, And so this does kind of go back to some of the important kind of just regular electrochemical considerations. So going back to the transmission cell, this is a good way to kind of start to think about how some of these requirements come into play. So for example, the optically transparent electrode, which is the working electrode for a transmission cell, is a very important component of the cell setup, because you have to enable both light transmission as well as fast diffusion to the electrochemical surface, because we all know that electrochemistry only happens at the electrode surface and nowhere else, right? So we have to have an auto setup that will enable quick diffusion to the electrode surface so that we can get that electrochemistry to happen fast enough so that we are able to detect our species in solution and eventually be able to, like I was saying, either deconvolute two different signals or get a pure signal from one particular analyte. And what that typically means is you are going to do like, often you'll do a controlled potential coulometry experiment so that you get full transformation from the neutral species or from the oxidized species to the Roux species or vice versa. All right, and so what this means is you're going to end up having these electrodes that are specifically designed to facilitate this fast diffusion and this facile electrochemical processes, and that's where the wire mesh electrode comes into play. All right, and so like I was saying, the wire mesh electrode, or you can also apply the same concept to the honeycomb electrode, but these wire mesh electrodes and the honeycomb electrode have very specific dimensions, in particular the width of the wires used in the mesh, the spacing between the wires, all are set up very specifically to enable these fast diffusion to the electrode surface. So if you see on this right-hand side, there's kind of a overview of a electrode surface where these striped rectangles are essentially profile views of a dissected wire grid, right? So the metal electrode is the rectangle, and then these kind of dotted lines demonstrate the diffusion layers as you move away in space from the electrode surface. And what this is showing us is that as you get a smaller spacing and smaller wires, you can increase the amount of diffusion, the kinetics of diffusion towards the surface, and you create a more linear diffusion regime, which enables fast equilibration times with the diffusion layer and gets you to a more full transformation from the initial species and the redox generated species quicker. And that enables us to envision, or I guess I should say, characterize faster processes in solution and see faster transformations. So the wire mesh, so kind of the requirements are, we want the wire mesh electrode to allow light to pass. We want it to enable facile diffusion to the electrode so mass transport can be neglected. And our equilibrium can be established quickly and then pushed towards either the oxidized or the reduced species by the potential. We want a thin solution layer. We want the cell thickness to be thinner than the diffusion layer so that the mass transport can be neglected. Again, we're trying to get the diffusion of the species to the electrode surface as fast as possible so we can build up the concentration of the oxidized or reduced species and then get to see the spectral signal of those species grow in over time. Right, so that is another reason why the cell is actually has this funny kind of bump in it. So we've got quartz over here, and then we have this like very small path length. Usually we have a 1 centimeter cell. Now we go between like a 2 millimeter to 5 millimeter path length to try to beat mass transport and enable our diffusion layer to be thinner than the thickness of the cell. And again, that's just to get our equilibrium to be quickly established and then pushed towards the product we want. All right, so some of the barriers that exist then in these experiments are, because we have such versatility, while this is actually a boon to this field, it's also somewhat of a barrier because you have non-standard cell design. And so that makes it a little more complicated sometimes to make sure that you're getting the best setup for your application. And you also need to make sure that your integration time between your instruments is appropriate to get qualitative and quantitative outcomes that are precisely measured for very fast processes. So basically, we are trying to make the integration time of the instrument faster than the process that's being measured. Of course, we want to be able to differentiate one signal from another, and we must be able to sync between the spectral and electrochemical instrumentation such that we can get real-time data. And that often happens, like some of this magic happens in the synchronization between the instrumentation. And in the past, there have been a lot of older home-built instruments that, you know, in the beginning of the spectral electrochemistry boom, people were trying to build these different systems, but they were having a hard time getting these measurements to synchronize to get this really precise measurement and get this real-time data, which made the data handling a lot more complex. And it made it difficult to synchronize the measurements and get really precise, accurate data. So recent kind of progress in this area is the availability of more commercial cells for a wider variety of different spectroelectrochemistry techniques. So at this point, there are commercial cells for not only UV-Vis, visible or UV-Vis, IR, Raman, but there's also commercially available cells for EPR, NMR, and several other different types of techniques. And one of the benefits of using a commercially available cell is that there are engineers in there that are working out all of the integration time issues and synchronizing the measurements between the two instruments so that they talk easily, and you can get a selectable integration time, getting that high precision and making your data handling a lot easier and making it a lot less difficult for the user to get the data they need and study the chemical application that they're looking for. And so kind of to follow up or to wrap us up with kind of the final, what would you see from the data in a transmission UV-Vis spectroelectrochemistry setup? This is one example, and it's from my graduate work, but I had it, so I figured I'd use it. So you end up getting, you know, two pieces of data. You get, especially if you do this as a controlled potential electrochemical measurement. You can also do like a cyclic voltammetry type of measurement, but in this case, we did a controlled potential coulometry, and we stepped the potential to about 100 millivolts past our E1/2 to oxidize our donor. And what we were trying to do in this situation is we were looking at this donor chromophore acceptor where we had light come in and excite a copper 1 center, which created an excitation to a metal to ligand charge transfer state, which then triggered an electron transfer out to an acceptor. But the tricky part of this complex is that the copper 1 is it is now difficult to then oxidize further, or sorry, re-reduce and then oxidize the donor, because these it's hard to find a oxidizable donor that has a redox potential that is competitive with the 2-1 couple on the copper. So we were trying to determine with our spectroelectrochemistry, are we able to oxidize the donor before any loss of the metal to ligand charge transfer state, and to make sure that our copper is staying in the copper 1 state and not getting reoxidized or oxidized further to the copper 2, because that is what we needed for our setup to work appropriately. And so we were able to see this by watching this this signal from our absorbents as we were oxidizing our donor, and we saw that before we had any oxidation of our copper, we got 2 of our donors oxidized, and that was stoichiometric with the actual complex. And we were able to see that in our absorption data, and I realized right now that I just pulled the wrong graph, so apologies. This is a different type of, this is not the coulometry graph, this is a different plot of the change in the absorption spectra. But this was the outcome, is that we were able to correlate these changes in absorption to the changes in the concentration of the donors and the copper, and that's how we were able to relate it back to the stoichiometry. So while I have 2 absorption graphs on here, you also get a coulometry graph, which would tell you how many electrons have been passed through your system, and that helps you also to verify the stoichiometry. All right, and so to take home messages, basically spectroelectrochemistry is a combined technique that combines spectroscopy with an electrochemical process. It's useful for observing in situ electrochemically generated species that are hard to isolate, synthesize, or are unstable. We can verify transient species with spectroelectrochemistry. We can probe electrochemically induced isomerization. We can elucidate reaction mechanisms. We can visualize single molecule behavior, and we can do so much more. Your imagination is kind of the limit here. However, the instrumentation is not standard. There have been advances in this area, but the instrumentation does depend on the spectroscopic technique and the electrochemical phenomena, as well as the sample specifications that will enable both your actual chemical phenomena to occur in solution while doing both the spectroscopy and the electrochemistry. And you need to make sure that your instrument response time and your integration time is appropriate to actually capture the kinetics of the process that you're measuring. In transmission techniques, the OTE, the optically transparent electrode, is necessary so that you can see, so that you can actually detect the light that is being transmitted through the solution. And it is an example of the way that cell design impacts the way that we actually, how we actually can apply these to different types of spectral and electrochemical techniques. And so with that, I just want to acknowledge, thank you so much to Dr. Ritesh Vyas and Ms. Nicole Navratil for inviting me to do this spectroelectrochemistry webinar. I love this topic, and I've been very delighted to come and share my interest in this with you today. I'd like to thank my colleagues and my collaborators and my research team at MSU Denver, as well as my funders. And I'd also like to promo a workshop that we are running this summer at MSU Denver. And if you'd like to learn more about this fascinating technique and get some hands-on experience with some of the instrumentation, I would love it if you would join us this summer. We're going to have a registration page up soon. But if you want to be notified, you're welcome to follow the QR code on the screen to add your name to a list, and we can notify you when the registration page is open. And so with that, I will turn it over to Dr. Vyas, and I apologize that I'm very far over my time.
Thank you very much, Megan. once again, really thankful, Megan, for joining us on this really wonderful topic. And moving further with the application part of spectroelectrochemistry, for me, the important thing is also the education and awareness about all the spectroelectrochemistry applications in modern world. um, we understood how spectroelectrochemistry fundamentally is very useful in variety of applications, but getting a holistic picture is also very important. So what I'm trying to do is trying to give like almost a project review or a literature review on where spectroelectrochemistry has been powerful in last 3 to 5 years. So we will cover some of these applications in DSSC solar cells, biosensor applications. We want to see how solution-based and thin film spectroelectrochemistry, they have diversified and have created an impact in modern science. How like Megan mentioned, not just UV vis, but what about UV vis NIR? How powerful that is as compared to ATIR or even Raman or even fluorescence spectroelectrochemistry. So the first application that comes to mind, obviously, in terms of light and in terms of how spectroelectrochemistry kind of developed after 1960 was solar cells. Back in 70s and 80s, DSSC was big. It is still big in terms of comparing against perovskite solar cells. So a big issue in solar cell, especially dye-synthesized solar cell, has been always understanding what kind of organic dye has to be used on P-type or N-type semiconductors or on those type of solar cells. And half-life of those dyes is extremely important. So we want to see the excited state of the dye, and the way to do it usually has been understanding homo lumo analysis, understanding the oxidation and the reduction curves. However, spectroelectrochemistry really kind of hits the mark in immediately understanding if this dye is going to be useful or not. How we do it? Say for example, a novel organic dye is in your hand. You can do a oxidation of the compound. Usually, it will be a bathchromic shift. You will have this kind of yellow to green color change, and the way you do it is you simply record. You record UV along the cyclic voltammetry curve, and immediately as the oxidation begins, or say you hit the oxidation peak, you're gonna see those curves that are corresponding to the oxidation or the radicals that are actually coming out, and you will immediately understand how stable those radicals are as you prolong the reaction. So in this particular case, what people generally achieve is they actually do a chrono-based experiment where they simply understand those voltages, and then they actually use those voltages and simply hold the potential for that 30 seconds and see what happens to the reaction. Obviously, if your reaction is actually emitting light, then it's going to emit those necessary dedicated wavelengths, and those dedicated wavelengths add those color change, and you will immediately see how stable your organic dye is. Now, something like this has been powerfully done with a fully built, like Megan mentioned. What you want to see is synchronization between potentiostat and the spectrometer. There are many companies out there. I'm not here to sell the product. What I'm trying to say is what you're looking for in terms of instrumentation is you want to have a complete package which allows you to synchronize the potentiostat and spectrometer to a millisecond level interval. Moving further, what we do offer are variety of kits. We also have a published article. You can get this presentation later on, and you can actually see how PalmSense has made a great impact in Spectro EC world, and there are tons and tons of papers that have been published on design, synthesis, and application of organic dyes. Moving further, I will quicken up some the pace so that we can have some discussions. In the biosensor research world, an important thing is using screen printed electrode to generate or say detect novel biomolecules. One of the very important study molecule in neural electrochemistry is dopamine. People study them for understanding what is dopamine oxidation current, or how is dopamine oxidation at higher and lower concentrations. What happens on a chain reaction of dopamine oxidation process is, on a lower concentration, it will provide a dedicated UV-Vis peak, which is corresponding to DAC, which is dopamine echrome. And similarly, for a higher concentration, it's going to dedicate a dedicated UV-Vis peak, which is corresponding to DAQ. And the best way to do this is you can actually combine it with spectroelectrochemistry, apply the same oxidation potential, just simply vary the dopamine concentration in the cuvette, and you're going to see the magic. And that's what this paper is about. They've actually created a spectroelectrochemistry based sensor which can detect dopamine, and they have actually tested in real world samples as well. We do provide a screen printed electrode based setup where you can actually have fully built plug and play system as well. And there is a dedicated reference article using PalmSense for Spectro AC. And again, I'm sharing this presentation so you can always go back and refer how it has also been used in the detection of anti-cancer drug gemcitabine and in real world serum samples as well. Moving further, if you are talking about spectro AC, you have to talk about electrochromic windows and conducting polymer, because there are tons and tons of conducting polymers who are very, very useful in modern day science in electrochromic applications. So I've selected this paper because it really clearly shows how thin film spectroelectrochemistry is useful. So moving from solution based to having a thin film on an ITO slide, we do provide a dedicated flow cell based setup where if you want to develop a electrochromism based optical sensor, which can be further used for something like copper detection or multiple trace detection, then this is where you can actually combine our Spectro AC kit, which with a cell setup such as this, and you can have a lot of electrochromic studies as well. Many people don't know that Spectro EC has been very powerfully used in understanding thin nanoparticle deposition. Because nanoparticles, they are UV-Vis active, you can actually monitor the deposition, not just electrochemically, but also spectroelectrochemically, using spectroelectrochemistry as a mean to monitor a thin nanoparticle deposition as well. There are tons of reference articles published with PalmSense as well. We have selected that over here. Moving further, going into deep dive UV-Vis NIR-based material synthesis, I have selected this paper. As you can see, the range now is not 200, it's all the way from 300 to 2100. It covers the entire UV-Vis NIR region. It's very powerful if you are looking into developing a new organic based molecule, or if you are trying to synthesize some new organic materials for electronic materials, with very high conductivity. This is where the application reaches. We do have the same package, we do have the options. There are other players out there. The only difference is, my suggestion as one of the experts here, select something which is UV-Vis active and ask the vendor if you can upgrade the same UV-Vis active to a NIR region. That will also save a ton of money. And guess what? If it is an expandable system, then it's really great. You can do a ton of work, with UV-Vis as well as NIR. Moving further, this is my favorite topic. This paper is also listed on BASI website. I really like this paper because it kind of covers how spectroelectrochemistry can be used in electrocatalysis world. They've used ammonia oxidation reaction as an example. This is in Journal of Catalysis, but they have targeted not one, not two, but actually 4 different spectroelectrochemistry technique. They have mentioned all the details on how you should set up, how you should trigger. They've combined PalmSense with Avantis, and they've used programming scripting as well. They are also kind enough to share their script on how to trigger the spectrometer. It's really a very good reference article. So in this paper, they have first started with UV-Vis. They've understood the air saturated and oxygen saturated electrocatalysis with UV-Vis. Then they have used this exact same setup, but then they followed that one up with Raman spectroelectrochemistry, in situ Raman. Again, they've used our setup, and then they have used a really nice, their cell is a homemade Raman-based Spectro EC setup, but for us, we are providing a very highly customizable Raman Spectro EC setup as well. We'll not go much into the details because this is a basic level session. So it's a ATIR, IR based Spectro EC setup, but we do have some really advanced IR based, NMR based, XRD based, transmission based, and even to some extent microscopy based Spectro EC setups. Last but not the least, fluorescence-based spectroelectrochemistry has also been really popular in modern days, especially in understanding or say separating biological matrices. And this is a very good example or a paper in Nature Photonics for understanding spectroelectrochemistry with fluorescence. Again, we do offer an expandable package in fluorescence spectro EC as well. And we also provide a lot of education. Say, for example, we do have a dedicated Spectro EC based application note, which is freely downloadable as well. For us as vendors, it's also very important. Again, we are not here just to popularize our message. What I'm saying here is if you are working for a Spectro AC setup, make sure you ask these kind of things from the vendors. You want to ask installations, you want to ask trainings and warranties about the setup. You also want to ask if your setup is glove box friendly. Not all setups are glove box friendly as well. So this is also an important reminder that if you are trying to purchase Spectro AC setup, do ask your vendors if they are glove box friendly as well. And that's my take on this Spectro AC from instrument experts point of view, and also from the point of view in terms of do's and don'ts in Spectro AC. Once again, thank you very much everyone for showing up for this webinar. We officially stop the session here, and we are open for Q&As now. Thank you.
Oh, thank you both so much for this wonderful session. We're gonna answer a few questions that have come in. And if you think of things, please feel free to put them in the Q&A, and we'll get them posted as they come in. But we'll start. I think this one is for Dr. Lazorski. So you discussed barriers to using spectroelectrochemistry. What is the biggest misconception electrochemists have when they first attempt to integrate spectroscopy?
Oh man. Biggest barrier. Okay, can you just repeat the final question?
Biggest misconceptions
Okay,
as they first get started.
Yeah. I think that my answer might be a little biased by my own research, but I'm guessing that likely the biggest misconception is that the electrolyte won't be an issue. Because sometimes, you know, there's a lot more interactions with ions in solution and with other complexes than I think we previously appreciated maybe 5 years ago. And having electrolyte in solution can exacerbate those issues, particularly when you're doing some sort of photoexcitation, because you're creating, you know, and obviously when you're doing a oxidizer or reduction, you're creating a charged species, but also you can create radicals and things like that that then can be stabilized or destabilized by the presence of additional ions. So I would say that being judicious and about the choice of electrolytes and being careful about trying to determine if there is any kind of interference that's happening because of the electrolyte would be one of the things that I would say. Yeah.
Awesome. And maybe this is a good follow-up question. How steep is the learning curve for labs without dedicated spectroscopy? I have a hard time saying it. Spectroscopy expertise.
I think that if you are not trying to go too far into spectroscopy, like hardcore spectroscopy and anything that has too much of a transient component to the spectroscopic setup. Like, for example, Dr. Bias was just telling us about how there are some new cells that they have for integration into ultrafast laser spectroscopy. That would be jumping off the deep end into the sharks, right? But if you're just trying to do ultraviolet visible spectroscopy, I think the learning curve is relatively small. I think you can get set up for that kind of thing, or near IR, kind of some of the things that some of the more common spectroscopies that you do in almost any type of lab. I think that barrier is pretty low to understanding because you've already got some background in the spectroscopy from just your general scientific education, as well as the background in the electrochemistry. So then putting it together is just kind of the function of just reading a bit and going into the internet and watching some videos and doing some self-study. But otherwise, I think if you're trying to go into the hardcore spectroscopies, time resolved spectroscopies, that stuff's going to be a little bit higher of a barrier. But starting small and starting with approachable techniques is a good place to start, and I think you can jump off from there.
Can I add a little bit to that, Nicole? Any vendors right now, modern day vendors for spectroelectrochemistry, I think all the top vendors, they provide dedicated trainings. You want to look out for vendor. I think Basi obviously is one of them who can provide you a 2-day course on Spectro EC, especially just with UV-Vis. The integration is not difficult. The do's and don'ts are more related on your own complex, where how much homework you have done in making sure that you have individually done successful electrochemistry experiments on your sample, along with, even if it's individual UV-Vis experiments. Big thing is concentration, the diluteness of the concentration, or at what concentration you are actually looking at your reaction. And the more important thing is if the question is related to understanding the software and the triggering and understanding the two different instruments, modern day that is covered. You can get trained on understanding and triggering in, I would say, less than a week. You should have a successful spectroelectrochemistry experiment of your own sample.
Thank you. All right, we'll switch gears a little bit. I think this one is for you, Ritesh. How do you account for solvent and electrolyte effects when correlating redox potentials to photovoltaic performance? Hopefully that makes sense.
Can you repeat that one more time?
How do you account for solvent and electrolyte effects when correlating redox potentials to photovoltaic performance? Voltaic.
So it's a two-part question, right? You want to make sure that, I was just talking about that in terms of concentrations and dilutions. what you want to see is, Megan very nicely explained it in her presentation, that your electrochemistry is not running slower or faster than your spectroscopy. What you want to see is, from your solvents and from your dilution of the solvent, you are getting a clear enough response from oxidation to reduction, and there is no overlap of the signals. In other words, the 100 millivolts thing that she mentioned. There is also, people ask us, why is that? Because if even if you are triggering both spectroscopy and electrochemistry at the same time, you want to make sure that the actual trigger and actual data acquisition is simultaneous, synchronous on millisecond level. Not all potentiostats actually do have that kind of sensitivity. So it also depends in terms of potentiostats and having those measurements done on that level as well. Megan, if you want to add, I mean, you really nicely explained in your presentation.
Um, I think that's a pretty complicated question, because if you're thinking about photovoltaic performance, I mean, I guess you're thinking about dye-sensitized solar cells if you're thinking about solvent, right? But I think, I mean, aside from what you just said, Ritesh, where you're thinking about the response function of the instrumentation, the kinetics of the reaction, but also solvent effects on redox potentials is like kind of an experimental issue. So you would really, I think, have to run a lot of a strong set of controls to determine like how your redox potential changes with respect to solvent, and what solvents you're running your photovoltaic in, to see like if there's a correlation there. But that is, I'm not sure if that's really answering the question that you're asking, but that is what I would say. Because it would just be down to really understanding the system very well, and understanding how that impacts the electrochemical potentials. And then, of course, if you have certain types of transitions, you can use the Rem-Weller equation
Yep.
to determine what the oxidized or the redox potential should be in the excited state, which will influence your performance as well.
Now, also remember, see, you are doing electrochemistry, and electrochemistry, you are getting a 10 ml volume in a beaker, or a 20 ml beaker. You are getting this nice little oxidation reduction, the duck-shaped curve, or cyclic voltammetry. Cuvette, the volume itself is 1 to 2 ml. So one thing I always suggest my customers is, repeat what you are doing in 10 ml cells. Repeat that first. Don't do spectroscopy. Just repeat that in the cuvette, just with the cuvette cell, the hotel cell. Don't do spectroscopy, or reverse it. Just do spectroscopy in that cuvette and see, first of all, you are getting something without electrochemistry. So that takes care of the dilution effect, that takes care of the solvent effect. Why? Because it's not the first shot technique. You have to work to manage that control and optimize your measurement. And yes, once you kind of optimize what you are getting in cuvette, you will start getting results.
Well, thank you. So here's another question. If I want to measure at sub-zero temperatures, is this instrument able to work in those conditions?
That's a very good question. So there are very few companies in the world. Avantis, with whom we have partnered, they actually do offer low temperature cells. They are spectroscopy cells for low temperature. We are trying to build something combining those low temperature cells with electrochemistry. because if you look at our website recently, we just recently launched airtight cell, which can be used at low temperatures. And that is the overall point of going in that direction, because a lot of electrolytes, in the modern world, they like the low temperature measurement. We are familiar with that. So the correct answer to that question is, you can individually contact us and we can create a project and we can see how far we can go.
All right. And here's a
Can I add a
similar
little
one.
bit on
Oh,
to
yes,
the low
absolutely.
temperature one? I think, like, well, clearly the limitation here is the instrumentation here doesn't even, getting it to exist. But then you also want to think about like the other material properties, I think, because I think there's another component of spectral electrochemistry that makes it really exciting, is that you're combining a ton of things, right? And so if you're doing a low temperature experiment, you want to think about, like, if you're undergoing a phase transition, if you're in solution, are you undergoing a phase transition? Does your solution expand? Does your solvent expand or contract when it freezes? Because you also, you're going to have electrodes in there, and how will that impact, how will the expansion impact the or contraction of the solvent then impact like the fact that you have electrodes in there? Will that end up like cracking your cell or anything like that? So just, I would say, being careful and observant about like what your sample does at those temperatures. I'm guessing that you probably know that already, but remembering that like physics doesn't change when you have a new experiment that you can run, right? Like, so just trying to think about how those things would impact the overall experimental design as well. That makes sense.
Yeah, and okay, here's one that might be related. Like, all the case studies and cell designs were about low temperature applications. So we'll look at this a little bit differently. Do you also have commercial spectral electrochemical cells for solid oxide electrochemical applications?
That's for us.
Yeah, that's for you.
Yeah. So we are working on that. We have actually 3 or 4 new spectro AC cells that we are working on this year. Solid oxides is one of them. You want to contact us, and if a CDA is required, yes. If not, then you can tell us the details about your experiment, exactly what you are looking for, and we can definitely see into doing something there. I can openly disclose we are working on something where folks in batteries and battery research world, they would like to understand the lithium-ion insertion and disinsertion process, and I'm very heavily involved in that project. That I can officially disclose that, yeah, that would be one of the cells we are very actively working on.
Awesome. Okay, here's one I'm gonna pose to both of you. When designing a spectroelectrochemical experiment, what do you prioritize first? Electrochemical fidelity, optical sensitivity, or temporal resolution?
Oh, shoot. I think that's pretty hard. Like, personally, I think it's hard to like kind of make those into
Yeah.
a hierarchy without knowing the system. Because it's like, which one is the most important to your system, right? Like, if the most important factor for your system is that you have really fast kinetics, then you're going to have to go with temporal resolution. If the most important thing to your system is that the echem is a little funky, you're going to have to like pay most attention to how you're setting up the echem. So it's really, I think, system dependent, but I think to put those factors into a hierarchy would definitely be related to the properties of the system, what solvents you have to be in, and how that particular system behaves under spectral conditions, electrochemical conditions, and what is the process that you're trying to look at.
I will go back to my PhD 15 years, 20 years back. My professor always said this beautiful thing, ask why 5 times in research. What is the problem you are trying to solve? Exactly pinpoint that problem. We were working on polyoxomatlates. Polyoxomatlates, they are used in 18 fields of science. You can quench the photoluminescence of a layer by layer assembly, a polyelectrolyte film, which we did. You can use it for electrochromism, which we did. It changes color when you give the light to it. You can use it for charge storage, which we did. Again, after storing the charge, they emit a completely different color. So uh, every single time we had to check different things. Sometimes we had to see the excitation state. Sometimes we had to see the transients, the fast kinetics. A big thing for me in Spectro EC personally is integration time, because I want to really, really see what is the half-life of my reaction. That's how I approach it, where I actually see, okay, if there is an oxide radical, let me see through electrochemistry and let me see how stable that radical is, number one. Number two, what is the distance between the two oxidation peaks? Is there an overlap that I can avoid? And how is my system behaving at various conditions of my electrolyte in terms of pH, in terms of solubility, and those all things matter. Why? Because the more I know about my system, the more I can implement and I can be experimental in my spectroscopy approach. But that's personally me. Again, it all depends upon the problem you're trying to solve.
So case specific.
Yeah.
All right, I think we have a really good question to wrap things up, and this one is for both of you. Where do you believe spectroelectrochemistry is currently underutilized, either in fundamental research or commercial applications?
I'll go first.
Yeah.
For me, spectroelectrochemistry has a huge presence in electrocatalysis. Right now, I look at electrocatalysis, and most of the work, as just for if I look it correctly, you're looking at ORR, CO2 reduction, OER, hydrogen evolution, because there's just lots and lots of dedicated work on all of this. What people forget is electrocatalysis has been always there for like 100 years. People have studied gazillion catalysis, a gazillion types of catalysts for variety of applications. Um, for me, if you can mix electrocatalysis using spectroelectrochemistry and solve the fundamental problems in a completely different science, something like biological science or something like enzyme microbial activity, then that kind of completes the picture where you are truly applying something like that. Um, a good example is a lot of machines that we see in the medical world, medical instrumentation. The electrochemiluminescence is a core in that, where a lot of big machines that you go out and you see and you check your blood or you check your blood sample and you say, yeah, and that is the impact one application can make in terms of going out and checking the blood for hemoglobin or checking the blood. All those big equipments that we are actually seeing out there, we forget that somebody out there in 70s and 80s used electrochemiluminescence, very simple science, and then he cracked down the code, just one application, and that's it. It stopped there. And my point is why? It is such a beautiful application. You can not just mix one, you can actually mix it in 3 ways or 4 ways, and you can have tons and tons of different applications in wide variety of fields.
I would say for me, I'm very fundamental, like basic science person. So, and I am fascinated by spin. And so I think one place that for me, I'd like to see, at least I'd like to see it grow is in application to EPR and NMR. I think there is a commercially available cell. It's been done, SpecEC has been done in EPR. But now we have transient EPR, right? Now we can pair light with EPR and make a whole other dimensional experiment. And I think that would be awesome because in particular, I think people are starting to see that that spin is so much more important to so many fundamental reactions and fundamental processes than we thought or gave it much credit for before. And spin states can drive like an enantiomer selectivity and just reaction selectivity, and do drive a lot of fundamental processes. And it hasn't, I think that's been like kind of underappreciated. And particularly with the fact that we have now transient EPR and light-induced NMR, those are two places where I feel like you could create a whole new dimension of SpecEC, where you have not only an NMR pulse, but you have a light pulse and you have an electrochemical experiment. So I think for me, it would be kind of manipulation of nuclear and electronic spins, and understanding that with respect to electrochemical and photo excitation.
Excellent. Well, thank you. Thank you so much, Dr. Lazorski and Dr. Baez, for sharing your expertise and your experience
Absolutely.
with us. Thank you to all of our attendees and those who submitted these very comprehensive questions and lovely discussion. And hopefully we will see everybody at our next webinar. Thank you again.
Thank you, everyone.
Thank you. Pleasure. Thank you.